And I'll be your host today. Before we start, I just want to remember that this meeting will be recorded, and I ask that everyone turn off all their cameras and microphones for a better video and sound quality. And after the presentation, you can raise your hands and turn on your microphones and cameras for the question discussed period. We happy to have with us today Dr. Zulma Vitezica. Dr. Zulma, she graduated from the University de Buenos Aires and got her PhD in Institut Nacional Agronomique Paris Gringon. She's a professor of animal breeding and quantitative genetics at Institut National Polytechnique de Toulouse, and her research includes genetic evaluation and genomic prediction in animal and plant breeding, in particular, considering dominance and epistasis. We appreciate to have you here with us today, and I'll pass it to you now.
Okay, thank you very much for the introduction. So I'm sorry for all the problems, the technical problems. And so I will talk about genomic prediction with non-additive genetic effects. So let me start with the plan. I will talk about which is a biological and statistical effect. Then we will see some about the new models that account for non-additive effects. Then we will see if they are useful or not. And then I will show you some conclusions. Okay. In the first part, we can say that if we talk about these biological effects, we can say that dominance and epistasis described apparent distortion of Mendelian segregation ratios. And so we have that because one allele or gene mask the effects of another one. And you have a very well-known example about dominance, and then you have a small example where we have two loci, A and B, and for being a unicorn, we need an effect and dominance by dominance to locus epistasis. Okay, but unfortunately, we don't know all gene actions and all the pathways. And for many purposes, we need to make some educated guesses. Why? Because we need to predict phenotype of progeny, because we want to know if some region in the genome is interesting, or because we want to know what happened in this genome region. And so for more practical purposes, we use statistical models. So if we talk about now these statistical effects, Fisher described the dominance and the epistasis as deviation from the additivity in a linear statistical model. And so we have to remember that dominance and epistasis are a phenomenon that are defined at the population level. And so we have the well-known genetic model where we have our phenotype, the additive or breeding value, the dominance deviation, and the epistatic deviation. Okay. Fisher decomposed the contribution to the genotypic value from a single locus and showed that this decomposition is a regression. And you have the regression here. And so you can see that the dominance deviation are essentially residuals. And this dominance deviation are the difference for genotype in red between the genotypic value and its prediction for two alleles. And the slope of this regression is the substitution effect of one allele. Okay, but we have this statistical effect. Why this substitution effect or alpha is relevant, and how does it take care of non-additive gene actions? So how we take care about the biological effects. The statistical definition does not care about how alpha works. And by definition, alpha potentially includes all biological dominance and epistasis. For instance, you have here an example with a pairwise epistasis that I took from, that is a slide from my colleague Miguel Toro from Spain. And you can see here that you have the substitution effect And in this substitution effect alpha 1, you have different biological effects. You have the additive, the dominant, and all the epistatic effects, additive by dominant, and so on. If we come back to the example where we have the unicorn, we can see that for being a unicorn, we need a dominance by dominance to locus epistasis. And this effect is biological. But this effect that is biological is inside the statistical effect that you have here, that is alpha 1, that is the substitution effect. So we have this both kind of effect that we have to keep in mind, because if we look, in addition to the effect, we look at the variance components, we will see that the additive gene action, that we have that here, contribute only to the additive variance. If we look the dominant effects, they are in the additive variance, but they are also in the dominance variance. And if we look at the epistatic effects, they are everywhere, in the additive variance, in the dominance variance, and also in the epistatic variance. So there is no correspondence between the kind of gene action and the variance component. Okay, now that we have all these maths, we can use it because in the absence of knowing true action of genes, all this math give us tools. For instance, the statistical additive effect alpha say, how much do you improve if you select me, for instance? The other is, if alpha is big, we can say, oh, it seems that it's an interesting locus. The other thing is that you have an statistical dominant effect. It says, for whatever reason, the heterozygote here is interesting. And we can say also that perhaps we can make these two individuals here and maximize it. In addition, if we have an statistical epistatic effect, this effect says somehow the fate of these two loci are bound together. So we have to keep in mind that this statistical additive effect is the only component involved in selection, because only the individual alleles are transmitted from parents to descendants. And the statistical dominant effect and the statistical epistatic effect also contribute to the total genetic value and to the expected phenotype of crosses of hybrids, for instance, but not for selection, because the allele or gene combination are not transmitted to the descendants. Okay, now let me talk about what we have as new models that account for non-additive effects. And we will see what is, we can have GBLAB, GDBLAB, GIDBLAB, and all these extensions. We will start with that. But before going directly to the different models, all these models can be used in animal genetic, but also in plant breeding. And I want to show you that we use that in animal and in plant, but the schemes are not exactly the same. In animals, we can use it in purebred. We can use that in crossbreeding. And in plants, we also use that in crossbreeding. For instance, when we will see that, I will show you an example from maize. And in maize, in general, we are interested in the hybrid. And this hybrid is generated by different lines, but one line is from one population and the names are heterotic group, and the other line is from another heterotic group, and this heterotic group are different. But the lines are fully homozygous. So all the F1 that you have here, that are derived from the inbred lines are genetically identical. We don't have Mendelian sampling. All the epistatic combinations that are in this inbred line are also transmitted to the F1. So we have to keep that in mind to understand one of the models, the last one. Okay. When we use this genomic selection and we think about non-additive effects in animal and in plant breeding, we will use that. We will use two approaches, but they are complementarity. One is focused on predicting additive effects in successive generation of a breeding program, because the objective here is to predict the additive genetic value to create genetic progress in the long term. We use that in purebred, we use that within these heterotic groups in maize, or within pure lines, for instance, in pigs. The other approach is interested in predicting the total genetic value of individuals to assess their performance or the commercial value. For instance, in purebred animals, but also in crossbred animals or in maize hybrids. What we can use for predict the different, for the prediction, we have two different way to work. One is the kernel-based prediction, and the other is the mixed model-based prediction. The kernel-based prediction is very similar to the blood machinery. Most of the kernel don't account for epistasis, because a kernel that includes epistasis should measure similarity simultaneously between pair triplets of loci across individuals. And I think that kernel, that is in my opinion, are good predictive machines, but they don't help to understand genetic progress and to make breeding plans. So for this reason, I prefer mixed model-based prediction, because we use the quantitative genetic theory to build relationship matrix that we know, and then we will put this relationship matrix into mixed models, and we will use all the things that we know. Okay. For genomic prediction with these non-additive effects, we need to construct a linear model, for instance, based on SNP genotypes. We will write orthogonal incidence matrices for additive, dominant, additive by additive, and so on, SNP effects. And this yields SNP lab or random regression lab kind of models, but they are cumbersome for epistasis. So we can do something that is equivalent, is to define relationship matrix. And we will do that in a way that high order matrices are products of low order matrices. So the whole theory, you have that in the different papers about additive. We have a paper of 2013 where we added dominance. Then we have another paper where you have any epistatic, we added the epistatic interactions, and then the last one where is the epistatic interaction. We consider that, but in a very special case for hybrid crops. Once we have all these relationship matrix, we put them in a mixed models, and we use all the things that we know. Remember that all individuals must be genotype or infer with certainty. Okay. We will see the first model, that is a model where we have the additive part and the dominant part. This is a classical model that is in terms of statistical value for breeding purpose. So what we have is the additive genetic value or the breeding value and the dominance deviation. But there are another model that it was proposed by Su et al. In 2012, that this model, I call it a genotypic model, because in this model, they use biological values at the gene level. Both models, the classical one, that is the model that you have in Falconer, and these models are able to explain the data, but their results and the interpretation are different. The classical model gives you breeding values and dominance deviation. The genotypic model predicts the additive and dominant genotypic value. The other thing is that the additive effect from the genotypic model cannot be used for selection, and the variance component that you estimate with this model should not be interpreted for breeding purpose and should not be compared with the pedigree-based variance component. So this model is useful, and it has been used for genomic prediction, but the results must be interpreted with caution. Okay. The models where, in addition to the dominance, we include the epistasis. One of the first approaches for defining an epistatic relationship matrix was developed by Jan and Rafe in 2015, but they only consider two second-order epistatic relationships, so it was only additive by additive. And also Martini works with this model, and they call the model the EGBLAB model. But none address the issue of orthogonality in the model and all the possible interactions. Their model can be used for phenotypic prediction in hybrid, but not to predict breeding value or estimated variance components. We resolve this limitation expanding the model, an approach that the name is NOIA, for natural and orthogonal interaction, that you can find in this paper of Alvarez Castro and Carbor in 2007. What we did our method used the NOIA approach, which builds the incidence matrices, but based on genotypic and not allelic frequency. The model is general for population, and we don't need Heidelberg equilibrium. So it's possible to use it in crosses or in homozygous lines. For instance, if you use that in inbred crops like in wheat, the NOIA approach gives breeding value for selection within the population. It was recently used in wheat by Raffo et al. And also in maize by these authors. Okay, the NOIA model can be used within hybrid, defining gene effects identically across parental groups. And that is a model that is very old, but because we can find it in the paper of Stuber and Cockerham from 1966. However, in that case, the NOIA model does not produce breeding values defining in each group. And for instance, in maize, the selection is within this kind of heterotic groups. So we need breeding values that are defined in these heterotic groups. And later I will present you another orthogonal model to obtain these breeding values. Okay, now this is the recipe. The first thing that we have to do is to define the incidence matrices. So we will define SID, that is here, and as you see, we don't use allelic frequency, we use the genotypic frequencies, and W, that is SID, is for the substitution effect, for the additive part, and W is for the dominant part. And so you have the definition of the dominant of W here, and then you have the definition of the different relationship matrices. Where you use the incidence matrices for SID for the additive relationship matrix, that is for the dominance, you use W. And when you have to define the epistatic additive by additive relationship matrix, you will use a Hadamard product of the additive, the genomic additive relationship matrix. And the only thing that you need is a standardization that involves the trace of these two matrices. And it's the same for the additive by dominance effect and so on. So that is possible to build all the different epistatic matrices with the additive and with the dominance relationship matrices. Once that we have that, we can use that in a mixed model. In the mixed model, you have the phenotype, you have the fixed effects, then we have the additive effect, the dominance deviation effect, and the epistatic effect. And you have to also consider a permanent effect, or here we call it the residual genetic effects, because this residual genetic effect will capture all the remaining genetic effects that we don't put in the model. Something that you have to keep in mind is that the matrices of higher order, so additive by additive by additive, or additive by additive by additive, are increasingly less informative, and at some point they are not worth fitting. So if you, even with a lot of data, if you are right to fit a second order epistatic effect, it's okay. So don't try to put, because it's very difficult to obtain good estimations. Okay. So we have a model where we can include dominance. It's possible to include also epistasis. And now we will see how we can use dominance and epistasis, but in a very special case that is for hybrid crops. And we will use the example for maize, where we have these two populations. And we have to remember that the NOIA model does not produce breeding value defined here in each heterotic group, but the selection is within this group. So we need to define breeding values. Okay. We did that with David González Diegues, that he was my PhD student, and he finished in 2021. David is a plant breeder, so I learned a lot of things working with him. And what we did is that we defined the seed matrices within each heterotic group. And we defined the W matrix for the dominance deviation effect in the hybrid. You can see that the recipe is exactly the same. So we will have an incidence matrix SID1 from the substitution effect for the population 1. You have that here, and as in the heterotic group, the lines are inbred lines, so we have this form. It's the same for the population 2, and then we define W in the hybrids, and you will see that in the hybrid we have the allele frequency from one population, but we have also the allele frequency for the other population. With these matrices, we will define an additive genomic relationship within the population 1. We will define an additive genomic relationship matrix within the other population, within the other heterotic group. And then we will define, with W, we will define the dominance genomic relationship in the hybrid. And with these matrices, it's possible to build all the epistatic matrices. But in this population, we have the possibility of separating the additive by additive intrapopulation epistatic relationship, and we have also an additive by additive across population epistatic matrix. And remember that it's possible because we have inbred lines. Okay, that is something that in plant breeders they use a lot, because they talk about the general combining ability and the specific combining ability. So they use a model that is according to this parental origin, taking into account the different heterotic groups. If we look inside this general combining ability, we can see that we will have the additive effect. For instance, for the general combining ability of the population 1, we have the additive effect of the population 1, but we have also the intra-population epistasis. And it's the same for the population 2. And when we look at the specific combining ability, we will see that inside we have the dominance deviation, and we have also the epistatic across population, the additive by additive, and then we have the additive by dominant, the with the first population, with the second, and the dominant by dominant epistatic. Okay, what happens if instead of having crops, maize, we have pigs? In maize, the phenotypes are in the hybrids. So assuming that we have only phenotypes in crossbred animals only. So if we know the faces, the model is identical. But if this model is also known as breed of origin of allele, the name is BOA. But if we don't know the faces, we can't partition the additive effects in the two populations, and we can't partition the additive by additive effects into intra and across population variation. So we could still use the NOIA model, but it's unclear how to obtain the EBV within the population. If we ignore epistasis, something that we can do is that we can backsolve SNP effects and obtain from that the EBVs. If we have phenotypes and genotypes, but in purebreds and crossbred animals, in this case, we can use the model that is proposed in Christensen 2014, but we have to extend it to dominance. But it's possible. Okay. In general, when we talk about non-additive effects, we talk also about inbreeding depression. Okay, why we talk about inbreeding depression? Because the theory of quantitative genetics attributes inbreeding depression to the presence of directional dominance. What that means is that we have a higher percentage of positive than negative dominant effects. And in this case, the mean of the statistical dominant effect is not zero, so we have it greater than zero. Statistically, this translates into a regression on a measure of inbreeding, of homozygosity. So you will have a model with, you have the phenotype, we have the fixed effects, you have here this measure of homozygosity, and then additive effect, dominant effect, and so on. How we can measure this homozygosity? In different ways. The more easy, simple way is using genomic inbreeding, and the other way is, for instance, if you want to use run-off homozygosity. If we ignore inbreeding depression, we will have estimate of dominance variance that we will be inflated. The other thing that's including inbreeding allows to finer estimates of EBV. Okay, so now we know difference model, we have all this nice theory, what we can do with all of that. So the first question is it useful? And so if it's useful, it is useful for having an extra accuracy in prediction, or can we use that, what we know about variance component? Okay, if we include dominance and epistasis in the model, in general, it does not increase the accuracy of prediction of breeding value. It's a pity, but it's like this. There are several authors that work with that in dairy cattle, in pigs, in sheep, in beef cattle, in trees. So nobody obtain results that say that dominance, the inclusion of dominant increase the accuracy of prediction. The only case it was, there are an exception that is allelo in 2016, but it was for it was in Holstein and for fat yield. As you know, we have the gut wine in this case, so it's a very special case. The other thing is that the inclusion of inbreeding depression increased the predictability of the model. And we found that in pigs, and recently Roth found that also in maize. So that in this case, that is interesting. Other thing that is that fitting non-orthogonal models or non-fitting inbreeding bias variance component estimation. I will show you some results about that. Here you have an example where we have pigs that as the data are from PIC. The trait is the litter size. We have 4,000 genotypes sold with 14,000 records, and we have more or less after quality control 40,000 SNPs. So we did a variance analysis, and we estimate, we did a Bayesian analysis, and we estimate the different variance component for 5 nested models. The model where we only use the additive part, then we added the dominance, then we added the epistasis. And if you look in this graph, you have the value for the different variances, and you can see that the additive variance is more or less the same in all the models, and so on for the other components, for the dominance variance and for the others. So we can say that the model is empirically orthogonal. Why? Because the variance component estimates don't change by adding an extra term. And in this analysis also, we look at the accuracy of the prediction, but the inclusion of the dominance did not increase the accuracy. We also look at the value of the different variance component, with including or not the inbreeding depression. And you have here the additive, you have here the dominance variance, and you have 2 models that are the posterior distribution of additive and dominance genetic variance for a model that is in orange, where we don't include the genomic inbreeding, and in the other model we include the genomic inbreeding. And you can see that if we don't include the genomic inbreeding, we have an overestimation of the dominance variance. That is not new, because if you look at this paper, the Boer and Hochschild in 93, we had exactly the same result with in the pedigree, when the analysis were based on pedigrees. Okay, now I want to show you another example that it was in mice. That is one of the papers of David's PhD. We use a publicly available data from the breeding program that is from the University of Hohenheim. And in this data, we had 2 heterotic groups with different lines. We have a little more than 1,000 tested hybrids. The trait was the grain yield, and there was, around 40,000 SNPs. And we used 2 models. The new GCA model, that is the model that is able to obtain breeding values, estimation of the breeding values in the heterotic group. And the second model was the no-YAM model, that here we call it the G model. So in this case, we define exactly the effects across the groups. In the GCA model, you see that we have the additive part for one population, for one of the groups, for the other group, the dominance deviation effect. We have the epistatic effect within the population 1, within the population 2, and we have the epistatic OC effect across the 2 populations, and we include these residual genetic effects. In the NOIA or G model, in this case, we ignore the origin of the gametes and use an effect that is the same that is defined in the hybrid. So we have an additive effect for the hybrid, the dominant deviation effect for the hybrid, and an additive by additive effect for the hybrid. In this case, we don't have residual genetic effect because we didn't have, we have only a single record. And the inbreeding was not fitted in the model because we did some preliminary analysis, and we showed that it was the homozygosity for all the hybrid was more or less the same. Okay, so what we did, we look at the difference variance component, the additive and the dominance and the epistasis, and we see that for the GCA model, this GCA model is reasonable orthogonal, and it's empirically because you see that we have more or less the same estimation for the additive variance. So these additive variance were very similar across the different models. The difference in the models is that we added more and more effects, and it was the same for the additive variance in the second population. And the other thing that we saw is that it's important to use the residual genetic effect or the permanent effect to accommodate all the non-additive effects of the lines that are not in the explicitly used in the model. What we saw when we compare this model with the G model, we see that the G model, even if it was constructed orthogonally, is not empirically orthogonal because you see that we have variation in the estimation of the different variance component. But if we look at the heritability, well, the values that we obtain with the two models were similar. Okay. So with this GCA model, we can say that it's possible to split the specific combining ability into a part of that is from the dominance part and also across group epistasis. And it's the same, we can split the general combining ability in one part that is from the additive effects within the line, and also an additive by additive epistatic effect within the line. Sorry, that is across the line. Here we have it's an error. Alpha, so the statistical additive effect, the dominant statistical effect, and all the epistatic effects are orthogonal. Orthogonal means that the definitions are given in a manner that a priori they are independent, so we don't have covariance. This gives a correct interpretation. And not, if you don't use orthogonal definition, you can have a spurious overestimation of dominance or epistasis variances. So it's important to use a model where you know that you have an orthogonal definition of the different components. We have to say that orthogonality is tricky in populations that are not in Hardy-Weinberg equilibrium. For instance, in maize, it's not very easy to have that. It is not always guaranteed because we have linkage disequilibrium, yes? So we have another problem, but it's important to work with the most reliable statistical model, such as an orthogonal model, even if we know that we can have problems with orthogonality. Other thing is when we think about variance component, it's important to take care about this variance component because you know that the genetic progress that we have by selection is proportional to the additive variance. So if we have estimate of the additive variance that are inflated, we will have an over-optimistic prediction of genetic gain. And on the opposite, if you have estimate of dominance variant that are inflated, we will have a high expectation of assortative mating, for instance, and when we have, for instance, two lies to produce a hybrid. So, and in this case, we have to take care about the estimation of the variance component. What are the practical implications of this GCA model? Is related to the breeding program, and that is especially for maize. Why is applying GCA into additive and epistasis within group is relevant for planning the genetic progress? Because we have to remember that the additive variance, instead of the total GCA variant, is more appropriate for the prediction of genetic progress obtained by selection within the heterotic group. And the variance of epistasis within the group will be converted in new additive variants by random drift and affected the long-term selection response. And you can find that in one of the papers of here. Why splitting specific combining ability into dominance deviation and epistasis across group is important for hybrids? Because the dominance deviation could be used to maximize this hybrid performance. So that is also very important. The other thing is that the two models, GCA or G-model, are equivalent in terms of predictability of hybrid performance. But the NOIA or the G-model does not provide additive or breeding value within the heterotic groups. The G-model cannot be directly used for selection of inbred lines within the heterotic groups, and the GCA model is appropriated for genomic prediction and also for variance component estimation. So the GCA model results are practically interpreted and used for breeding purpose. Okay, so finally, I want to say that now we have a comprehensive theory. We know how to properly define and estimate non-additive statistical effects. Inbreeding should be fit in the genetic evaluation models. Fitting dominance and epistatic effect is interesting to correctly appraise genetic variances. Dominance and epistasis is not difficult with markers. If we compare that with the models where we had only pedigree, and provide that is not difficult with markers that we have in animals and also in plants, because we have many individuals that are genotyped. And in our experience, computational complexity is not an issue. Model fits into computer, but convergence and accuracy are an issue. And if you put many parameters and you have little information, you have problems, okay? So we have to pay attention about that. Okay, if you are interested about that, we have a review that we did in 2018 with my colleagues Luis Barona, Andres Legarra, Miguel Toro, and myself. And we have also a chapter in this book that is from this year, I think, no, last year, 2022. All this work, I didn't do that alone. It was possible to do that because we have many collaboration with many people everywhere. You will see my colleagues in the University of Georgia, people from Argentina, from Uruguay, my French colleagues also that work in animal science, but also in plant science, some colleagues in Australia, and also in Denmark and in Spain. And some PhD students like David, Tao, or Johans. So finally, I want to ask you to thank you for the invitation, all the research agency for finding the different projects, PIC for the data because they were very, very useful, and also our bioinformatics platform for supporting all the analysis. So thank you very much for your attention.
Well, thank you for the presentation. Was well amazing, very informative. We'd like to thank you for accepting our invitation. So if anyone has questions, just raise your hands and turn on your microphones and camera and feel free to ask questions. So if anyone has questions, well, while we wait for brave people here, I do have a question. At some point, you say that include the effect of dominance and these other effects, they do not increase accuracy. So I was wondering, if it does not increase accuracy, why would we include them in the model? There's another reason?
No, I don't. It's. I think that it's. It was very difficult to use non-additive effect with pedigree. So when we starting to do genomic models, we say, why not? With genomic is easier because now it's possible to look at the heterozygous and to look at the really the genotype. So we say, okay, we will see if we can do or we can include this non-additive effect with the SNPs. It's easier to do that, but we don't have an increase in the accuracy, at least when you include these effects. I think that all the biological part of dominance and epistasis, we have that in our breeding value. So our model is robust. But if you include, that it was a surprise, if you include the inbreeding, in this case, yes, we observe in pigs an increase in the accuracy because the fit of the model is I think that is better. And recently they found exactly the same in, I think that it was in maize. Yeah.
I see. You say that the computer power is not going to change. It's very impressive that it does not change that much, that you only need kind of the same power to run the analysis.
Oh, I don't say that it's, I see that it's not a problem because we have platforms and we have possibilities to do a lot of things with very complex models. I don't say that it's easy to find good estimates. You need a lot of data and you need informative data.
Okay. Okay, so we have Audrey here. You can go, Audrey.
Hi. Sorry, just actually about this question that Samina just asked. I'm sorry, I may need to present myself. I'm Audrey, so I'm a previous student in CGIL, and I'm now working with Gregor Gorging in Edinburgh. Actually, it's on the computational cost of this kind of analysis. I was wondering, because you mentioned inbreeding and also non-additivity. When you said that the computational cost is not that high, are you speaking about inbreeding or non-additivity? Because
inbreeding does depend how you compute it. If you compute it as in a very easy way, so you look at the number of homozygous divided by the total, the frequency of the homozygous divided by the total frequency. So it's very easy to compute that. If you do that with ROH, it's more complex. So, but it's not something that it will take, I don't know, probably that depends the quantity of animals that you have, but you have that in a very easy way. It's not complex. Okay. And then when you use, when you have to build the dominance relationship matrix, to build the matrix is not complex. To invert the matrix, that is more complex. So in general, you have to parallelize that to do that in a good way. But then when you have that, you put that in a mixed model and you solve it with, I don't know, BLAPF90, for instance. And so it's okay.
Okay.
No, you don't need to do that, I don't know, for 1 week of computation. No, that is no, it's not my experience. Yeah.
Okay, that's good. Thank you.
Okay, so we have Kristen also. Once you can go, Kristen.
My question was kind of, the other aspects of animal breeding, which you can apply, kind of these methods that maybe would be more promising than prediction accuracy. Are there any other ways you could apply it that you know of? That would provide more of a benefit, like mate allocation, or maybe in crossbreds?
We try with male allocation, and the results were not, how do you say? We did that with David. It was his second paper, but the benefit of doing mate allocation in pigs, it was not really very important. And the other thing that discussing with breeders, they told us that it's not very easy to do that in practical. So, but we try to do that with mate allocation, but the results were not were not very good.
Yeah, makes sense. Thank you.
Okay, so if anyone has any questions, this is the moment, or it's already 2:30, so anyone? No?
You may want to read the chat. Bayode is asking questions.
Oh, sorry, the chat. Okay, so Bayori asked, when inbreeding is accounted for in the model, does it not highly penalize inbreed animals?
But if you have inbred animals, you have to take into account that. So I don't know, what do you mean when you say that you penalize? So inbreeding, it's something that we have in our animal population, and we have to take into account that in the model. If you don't take into account that, for instance, the estimation of the variance components will not be good. And so the I don't know, the genetic progress that you can expect, it will not be okay. So you have to consider that in the population, and we know that we have inbreeding, so it's necessary to consider that in the model. Yeah.
Okay, so he also asked, the relationship matrix we use already account for inbreeding. So are we not double counting for inbreeding if included explicitly in the model?
No, but because in the inbreeding that you use in the model is an inbreeding that will allow you to have to respect one of the hypotheses of the model, that is the expected value for the statistical dominance deviation effect is equal to 0. Because when we have directional dominance, we know that this is not equal to 0. So we put this measure of inbreeding to take into account that and to respect the hypothesis that we have in the model.
Okay, so Cassiani has a question also. So how can the use of data simulation help to study these non-additive effects?
Excuse me, how?
Can the use of data simulation help to study these non-additive effects?
Simulation dominance is we did some simulations with dominance, and they can help you to understand the how you say they can help you to understand what we can expect in real life. So, for instance, we did a simulation with a crossbreeding scheme with 2 breeds and crossbred animals, and we use dominance. But for instance, in the simulation, we use the values that we obtain from the real data and simulated. If you want to simulate epistasis, that is not easy. That is not easy because we don't know if what we have to simulate. I don't know an interaction between 2 loci, 3 loci, 4 loci, or that is very, very difficult. Yeah. Yeah.
Yeah, and then a last question from Sebastian. Is there any difference in using a simple approach versus a runs of homozygous based approach for fitting in breeding the model?
I think that when you use the run of homozygote, the ROH, it's a how you say it, a fine way to consider inbreeding. But in terms of the model and then the estimation of the breeding value, I think that it's the same. Yeah.
Okay. Well, we would like to thank you again for your presentation. It was absolutely amazing. And I want also to thank everyone that attended today the meeting. And I hope to next week we don't have a seminar, but we expect to see you on March again.
Okay. Thank you for the invitation.
You're welcome. Bye.
Bye-bye.